Hey everyone. So I think we're going to get started. My name is Katie Grogan, and it's my pleasure to welcome you to the final culminating lecture in our series, Advancing Health Equity with Medical Humanities. We've been running this CME series monthly since April and have had the pleasure of hosting some incredible speakers, giving talks on topics ranging from abolition medicine to disparities in infant and child mortality, to migrant health ethics, to LGBTQ health and the media, among many others. And I will go ahead and drop a link into the chat where you can view videos of the previous webinars if you want to check them out. This series was developed in partnership with New York University, the NYU Grossman School of Medicine, and the Arnold P. Gold Foundation. Our initiative is part of the Foundation's Gold Human Insights webinar series, and we want to express our deepest gratitude to the Gold Foundation, and especially to Pia Pine Miller and Ann Bruder for their support of this project. I want to also recognize my partners in the development of this series, Dr. Kim Adams, who is the Humanities in the World postdoc at the Penn State Humanities Institute, where her work focuses on public humanities and medical humanities, and Sharanik Bosu, who's a PhD candidate in English at NYU, where he focuses on economic thought, literary rhetoric, and public humanities. So the vision for this project was to take an interdisciplinary, collaborative approach to interrogating inequities and injustices in healthcare and society more broadly, drawing from the humanities, the arts, the social sciences, and medicine. And in the spirit of this collaborative framework, we thought it appropriate to close out this series with a multidisciplinary roundtable discussion. So tonight we'll be hearing from 4 experts on the topic of equity and diversity in clinical trials, taking us from historical inequities in medical research and data collection to ways to improve the diversity of research participants moving forward. In the interest of time, I will give very abridged introductions of our esteemed panelists, but you can visit the Gold website for their complete bios. So we have Dr. Arthur Caplan, the founding head of the Division of Medical Ethics at NYU Grossman School of Medicine, a luminary in the field of bioethics, an advisor to many national and international ethics committees, and the recipient of numerous honors for his work in the field, including awards from the National Science Foundation, the FDA, and the American Society of Bioethics and Humanities. We have Dr. Aisha Langford, Assistant Professor of Population Health at NYU Grossman School of Medicine, and co-director of the Clinical and Translational Science Institute's Recruitment and Retention Core. Her research spans cancer prevention, cardiovascular disease, and clinical trial participation in community-based settings, and she serves as associate editor for the journals Patient Education and Counseling, as well as Clinical Trials. We have Dr. David Oshinsky, director of the Division of Medical Humanities at NYU Grossman School of Medicine and professor in the NYU Department of History. He is the author of numerous award-winning books, including Polio: An American Story, which won both the Pulitzer Prize in History and the Hoover Presidential Book Award. His articles and reviews appear frequently in the New York Times, Wall Street Journal, and Washington Post, among other publications. And we have Julie Sowash, Executive Director of Disability Solutions, a US-based nonprofit working with companies around the world to build both best and first-generation practices for the inclusion of people with disabilities. The programs she's built have resulted in thousands of people with disabilities finding and growing careers, and in the training of nearly 10,000 people leaders to reduce fear and stigma around all types of disabilities. And Julie was recently recognized as one of the top 100 diversity, equity, inclusion, and belonging professionals by Untapped in 2022. So we can't wait to jump in and hear from these folks. Just a few quick logistical notes. We're going to ask that folks kindly keep their mics off. Please put your questions into the chat, which we'll be monitoring, and Sharanik will read them aloud during the Q&A portion towards the end. But feel free to drop in those questions as they come up. There's no need to wait till the very end. And as I said, we'll be monitoring it. And finally, we're going to put a link into the chat shortly to a very brief survey. We'd appreciate if everyone could take a few moments to give us their feedback, especially if you're claiming CME credit tonight. And with that, I'm going to pass it over to my colleague, Dr. Kim Adams, who has a short video from us from the Gold Foundation, and will be monitoring the roundtable. Thanks so much.
And I'm just going to share my screen quickly.
The best healthcare is compassionate, collaborative, and scientifically excellent. Thank you for being on this mission with us to ensure humanism in healthcare for all. This is a Gold Human Insight webinar produced by the Gold Foundation and its partners.
Hello, I'm Pia Pine-Miller, Senior Director of Strategy and Business Development for the Gold Foundation. The Gold Foundation created the Gold Human Insight webinar series to bring inspirational live and pre-recorded webinars to healthcare professionals and learners. Our 2022 theme is Renewing Our Commitment to Humanism. With the myriad of challenges facing healthcare systems today, including patient disconnection, clinician burnout, and even more challenges highlighted during this time of the syndemic of COVID and social justice issues, the Gold Foundation's vision of a better future remains more urgent than ever. Today's Gold Human Insight webinar is one in a series that is part of a special collaboration with NYU School of Medicine, which is an institution that is a member of the Gold Partners Council, a group of medical schools, hospitals, and healthcare systems that are leaders in humanism in healthcare. Thank you to our presenters and our audience for everything that you do.
Today's lecture is part of a larger series on advancing healthcare equity with medical humanities. These slides provide a description of the larger series and accreditation information from the NYU CME office. They are required for attendees seeking CME credit and are available to all who are interested through the Gold Foundation website.
Okay, thank you everyone for listening to that. And if you'll bear with me one more minute, I'm going to spotlight our presenters. And I'm going to welcome everyone to this wonderful webinar on diversity and equity in clinical research. And I will be moderating the Q&A and asking questions to our presenters. And we'll take questions from the audience towards the end of the hour. So the first question that we have is for Dr. Langford. And it's pretty straightforward. So what is a clinical trial? How does clinical research inform contemporary medical practice? And what indicators of human diversity are relevant in the setting of clinical research?
All right. Well, I will say clinical trials or a clinical trial is really a broad umbrella. There are a lot of different types of clinical trials. So, for example, most people are familiar with drug trials or surgical trials, but technically behavioral studies could also be considered clinical trials. There's clinical trials related to vaccines and genetics. So there are different types and topical focus of clinical trials, and they are in typically 3 different phases. And if you think about in the case of drugs, or even in the case of the COVID-19 vaccines, typically you do clinical trials to get evidence and information that an experimental treatment or intervention is better or more efficacious or efficacious at all than a standard of care, or in the case of like the new COVID vaccines, developing the evidence base that a new intervention is helpful and effective for most people. And so that's kind of the broad umbrella of clinical trials and what they are. I should also say that clinical trials also focus on different patient populations. So you have clinical trials that really focus on older adults. You have clinical trials that are focused on pediatrics and gene therapy. And so there's different ranges and types of people that we're looking for. Not all clinical trials have all age spans. For considerations for diversity, there's lots and lots and lots, which we'll talk about tonight. We classically think of sex and age, but over the last 20 years or so, we've talked a lot about race and ethnicity, and that's a tricky topic because that's a social construct and not a biological variable, which we'll talk about later. But other aspects of diversity also have to do with different manifestations of a disease. So for example, if you're thinking cancer, you'd want different types of cancer, different stages of the cancer, et cetera. So that's kind of a broad bucket. I think people typically, at least my family and friends, when I first started researching clinical trials, they thought, oh, those are things for people who are really, really sick and dying, and it's all drugs. And there's really a huge variety of that, ranging from clinical trials for screening and diagnostic, and then clinical trials to help support survivorship care or even palliative care and everything in between. So it's not just for sick and dying people. There's also a lot of clinical trials that need healthy volunteers.
Great. Thank you for that. And our next question, since you referenced the vaccine trials for the COVID-19, next question I'm going to pose is to Dr. Caplan. So we recently ran a large-scale accelerated set of clinical trials for the COVID-19 vaccine. What changes are taking place in clinical research due to COVID and Operation Warp Speed?
Yeah, well, like Aisha, that question will let me talk for about 4 hours, but I'll try to trim it down a little bit. We basically did not cut corners, but we introduced a concept with COVID vaccine studies that basically was built on the idea of a public health emergency, that we had a huge threat in terms of death, morbidity, hospitals being overwhelmed, and we had to find something that would slow the virus down. Particularly, we're talking now about the earliest days of COVID, when it was both highly lethal and very few people had been infected, and people were literally worried that 10, 20, 30% of the world's population might die. I mean, just an enormous fear. So in order to cope with that, what we did was we took the standard clinical trial model, which Aisha began to describe, but tested on animals, tested on a few humans for safety, tested on a few more humans to get your doses and do you give it as a oral vaccine or a shot or whatever. And then the classic clinical trial idea, which is test it against a placebo group or test it against the existing agent to see whether it's better. That process historically to produce vaccines anywhere from 8 to 12 years. So looking at this emergency, people said, we can't do that style of thing. Now, luckily, some of the vaccines that were under consideration, Pfizer, Moderna, were built on a platform. Some people have heard of mRNA. We can talk about that later. But the interesting thing is they'd already begun testing it in humans for SARS, MERS, and Zika. So it wasn't a cold start. So people said, well, we can lop off some time. We don't have to do all the animal studies. And maybe we don't even have to do the first in human safety studies because we've got some information. Okay. But the other thing that people realized was there are huge numbers of people out there being exposed. We can accumulate a lot of data very quickly. We don't have to do what normally happens in a clinical trial. People are running around hoping to get people to enroll and trying to recruit them and looking at different communities and seeing whether they can sign them up. Here, there were just infection was rampant and you could easily recruit people to try out vaccines because they were afraid and desperate. This was not anti-vaccine attitudes. So we did do that. We did it under what's called emergency use authorization, which had never been used that widely, but it basically said, enroll big numbers, see what the safety profile looks like, and look for surrogate endpoints to see whether that might indicate that you're going to get immunity. And decisions were made by the FDA and advisory committees to approve those vaccines. And remember, it took a year. It was from January 2020 to December 2020 that we got vaccines out and began to give them to healthcare workers and nursing home residents in the US. So that was pretty fast, 12 years to one. And that got a lot of people with diseases, disabilities, disorders thinking, you know, I have a crisis. It's not a worldwide crisis, but if I have cancer or depression or ALS or other terrible diseases, or my kid has sickle cell or hemophilia or spinal ataxia, all miserable diseases, why can't you do the same thing for my disease? And so that's what the change has been. And I don't want to take the whole evening talking about this, but it's shifting attitudes about we've got to go faster. We have a model of how to go faster with vaccine studies of COVID, and many patient groups and advocates are saying, let's apply that to drugs or vaccines or even devices for other terrible diseases. And we're getting a lot of pressure. It's ironic because some people are like, I don't know about those vaccines anymore. They seem to get approved too fast, which bred, as we know, both disinformation and distrust that we see around today. But many patient groups are not in that camp. They're saying, let's move quickly.
And so what I'm hearing you say is that the speed with which the COVID-19 vaccine was produced has encouraged a diversity of patient populations to seek similar treatment for their
Exactly.
for their.
By the way, you
particular
could see it.
concerns.
Some of you familiar with Alzheimer's disease know that a drug called Aduhelm was approved on, I'm going to call it weak evidence, not no evidence, just less than you normally expect, partly due to large numbers. And patient groups were lobbying, saying, you know, there may be some risk here, maybe it's not going to pan out, but let us decide how much risk we want to take for ourselves or our loved ones who are fading away due to Alzheimer's. And you did it for COVID vaccines, do it for us.
And so I wonder if we might ask Dr. Oshinsky to provide us with a bit of historical perspective on this. Since the COVID-19 vaccine trials did draw a great deal of media attention, like the polio vaccine trials in the 1950s, what can the similarities and differences between these two studies tell us about building broader representation of diverse populations into clinical research?
Sure. Before I get into polio, I'd like to just add a bit to Art's really good summary of this. What made Operation Warp Speed so extraordinary, and not only did we do it in warp speed, but basically it takes billions of dollars to get a vaccine from the laboratory to the market, and only fewer than 10% of these vaccines actually make it all the way. And what made Operation Warp Speed so extraordinary and so unlike the polio experience was that this was a public-private partnership, the likes of which we have never seen. Taxpayers poured billions of dollars into this operation. Pfizer was somewhat different, but Moderna, virtually everything Moderna did was financed by the public. These vaccine companies that produced were given full legal immunity. They literally could not be sued for any mistakes that were made. They were first in line for any product that they needed, which was extremely important. And I think most important of all, and different than any trial, is that there is no drug company, there is no vaccine maker that would ever begin producing the vaccine before the tests were complete. I mean, because if the test went badly, you just have to throw it out. What the government said in this case is do both at the same time, simultaneously. And what that really meant was that these vaccines were being produced before the results of the trial were out. And what the government said is if the vaccine works, we'll pay you for it. And then you can make your own profit selling it. If it doesn't work, we'll still pay you for it and we'll destroy the vaccine. So when you have production and testing going on at the same time, you can save a ton of time. And that is very important. Let me just go into polio very, very, very briefly. It's a childhood disease. It's a viral disease. It's a crippler. There had always been a belief that polio did not really affect Black children, that it was literally a middle-class white disease. And one of the things that the March of Dimes, this was all done privately. One of the things the March of Dimes did was basically to disprove this. Now, what the March of Dimes did do was they had this huge polio center for people who had polio in Warm Springs, Georgia, and they would not accept Black children. In other words, this was during the segregation era of the 30s and 40s, and the March of Dimes was not going to get caught up in that issue. But what they did do was to set up an equally important and well-funded facility for polio survivors at Tuskegee, and these were all for Black kids. Basically, the polio vaccine was tested on animals very, very carefully, but it was the stage 1, stage 2 human trials were done at what was called the Polk Home for the Feeble-Minded and Retarded. In other words, Salk just went in there. He asked the head of this school to basically test his vaccine, and he was given the go-ahead. So in other words, mentally challenged kids were just fair game in this situation. And then in the stage 3, they basically had the largest public health experiment in American history. More than 1.5 million children took part in this. No one knew whether this vaccine worked. No one was certain how safe it was, but parents had a sense of risk versus reward. And by that, I mean, they saw polio every day. They saw the leg braces, the crutches, the iron lungs, and they were willing to push their kids into line. It was a double-blind study. Much of the group got the real vaccine, some got a lookalike placebo, and there were observed controls. Black children took part in this in numbers equal to their population. In other words, what the March of Dimes did was to take 200 plus counties where there's a lot of polio among children, and all kids in grades 1, 2, and 3 were basically part of the process. They either got the real vaccine or they got a lookalike placebo. It took before computers, it took a full year to analyze these results. And when it came down, the vaccine was safe, it was potent, it was effective. And that, along with the Sabin vaccine, essentially ended polio in the United States. It was an absolutely extraordinary event. And it was an event done in such a way that Black children got basically the same rights in this trial as white children, but in the roll-up, and certainly in stage 1 and 2, a lot of it was done on children who were considered feeble-minded or retarded.
Great. Thank you for that answer. And your comments on the COVID-19 vaccine process made me want to turn right away to our question on funding, but we'll get there. Before we get there, I have a question for Julie Sowash on demographics. So what demographic groups are most frequently represented in clinical research, and which groups are least frequently represented? What about outside the USA? What are some of the potential benefits of a more diverse sample in a given trial?
That's a lot of questions all in one. Let's see what I can get.
You don't have to answer them all.
Thank you for that. I appreciate that, Grace. I mean, I think we can tell by the conversations we're having today that traditionally clinical research has been geared towards white, able-bodied men, now including women. And when convenient, tested against underrepresented communities in an unethical way. I think the challenge that we have now when we're thinking about demographics is what demographics information do we want to collect, and what is the purpose of that demographic collection? So when I think about a process, I'm thinking really in these terms in 2 separate ways that we want to think about demographic collection. The first is what is scientifically necessary that's going to help us drive better medicine for a reflective population that is, reflective of the disease state in the therapeutic area. The second piece is really where my expertise lies, and it's how do administrators and sponsors and pharmaceutical companies take demographic information that's collected and identify ways in which to create a more inclusive and functional process where more people are able to complete those clinical trials. And so that can be, we start with always the big ones: race, gender, ethnicity, disability status, sexual orientation. We start there, and then we can even get further into marital status, socioeconomic background, education, those kind of things that will give administrators and frankly, business leaders the best insight into what information should be gathered so we can create trials that are both more efficient, more reflective, and also create that strong business case for diversity. And I think that's really, if I think about what we're missing right now, is that we don't have a good idea of who's participating in clinical trials outside of white people. And that's because we don't have consistent data collection requirements from the FDA or even just in best practices as clinical or healthcare professionals about what that data is when that data is going to be collected and what those common definitions are. And so what we do know is that in 2019, the FDA did a review of FDA oncology drug approvals done by JAMA over the past decade showed that only 63% of participants had race information gathered, race and ethnicity information gathered. That's a pretty low percentage to know about what the actual participation rate is. We also know that in 2022, Harvard MRCT did a study of all open trials on clinicaltrials.gov and found that most of the time, a vast majority of the time, people with disabilities were auto-excluded from participation. And so I happen to have multiple disabilities. They're hidden disabilities. And in almost every case, I would be excluded from participation in a clinical trial if I disclosed that disability, it was part of my medical history, or my medications were reflective of what that diagnosis is. And so really, the best answer to your question is we don't know, because we don't ask the right questions and we don't uniformly collect the correct data, and we don't create informed consent processes that allow us to share that data across and really understand what participation rates look like.
Great, thank you for that answer. And what I hear you saying is that we need more consistent data collection in this process, which I think would be hugely beneficial. Maybe let me address a related question about the structure of clinical trials to Dr. Langford. What is the distinction between phase 1 and phase 2 and 3 trials? And how does demographic representation, what we know of it, vary across these phases?
Okay, so phase 1 studies typically are small or smaller. They tend to be 20 to 80 people. In most cases, they're focused on healthy volunteers because you're looking at safety and dosing and things like that. If in a phase 1, if it shows promise, then it'll move to a phase 2 where you're looking at, again, larger safety, more efficacy. In a phase 2 trial, and again, it depends on the disease, but it can be anywhere from, say, 100 to 300 people. And if that still shows promise, then you look at the intervention in a larger number of patients, and that could be hundreds or thousands of people. And really the goal there is really to, often in a randomized controlled trial type way, you want to see if whatever you're testing is better than the standard of care. Or in the case of COVID vaccines, you want to see, are the vaccines actually helpful and resulting in fewer disease and helping protect people? So that's kind of the broad ways that you move through the different phases. So for example, I was actually in a phase 1 trial for the COVID-19 vaccine, and I was one of 48 people at NYU Langone. And my family thought I was crazy. They're like, why are you doing this like first in humans? What if you get hurt, etc. So that's really the kind of the broad progression of the phase 1, phase 2, phase 3. I will say in other diseases like cancer, the inclusion criteria for like who would be in a phase 1, phase 2, or phase 3 may be different and disease specific. What was the second part of your question? Sorry.
No worries. The second part of the question was about how does representation of different patient populations vary across these phases?
So there's not really an easy answer. I think it really depends on the disease. I think it also depends on where the study is being conducted. For example, many studies are conducted in large academic medical centers for a variety of reasons. Many are good, some are less good. And so if you think about, in general, who in the world goes to large academic medical centers, there is racial and ethnic diversity, but many folks, for example, of underrepresented backgrounds may be more likely to be seen in a community hospital. But typically community hospitals, solo practice, physician offices, and like federally qualified health centers, which are really, really an important aspect to our health system, often don't have the infrastructure, often are small, and therefore are not included often in our publicly funded clinical trial infrastructure system typically. And as you can see, that would be a huge mismatch of where the majority of the people are, but maybe not where the majority of the infrastructure for clinical trials are. So it's not an easy answer. I will say that there are many groups that have had a lot of success recruiting underrepresented populations. So I don't want to give the idea that it's impossible, because it's not. I've had several colleagues here in New York City and at other places. And if you can put intentional efforts behind it and be inclusive with your protocol and inclusive of your partners and inclusive of the sites, it's very, very possible. And it has been done. It continues to be done. But it really puts the onus on researchers, and it puts the onus on funders to maybe expand our thinking, beyond academic. I'm at an academic medical center. I'm glad there's like big funding there. Very grateful for that. But we have a big country. It's harder sometimes to get a clinical trial, for example, in a rural area. So we don't always talk about geographic diversity, but what if you live in the middle of like Nebraska or Appalachia or whatever, you have to travel 3 hours to go to a primary care visit. Like that's a huge need. And I don't think we necessarily talk about like rural health enough and how we really expand access beyond like big urban centers.
Yeah, and maybe following on that question now would be a good point to ask about funding. So I'm going to direct this one to Dr. Caplan. Do the funding structures for clinical research in the US reward diversity in patient populations or recruitment practices, and are and should subjects be paid to encourage diversity?
Well, let me go to the last point first. It's not clear that you're going to get more participation by paying people. It might seem intuitive that you would, but some people look at it and say, if they're paying me, must be pretty risky. I don't know that I'm going to do that. So you have to be a little careful about financial incentivizing. As Aisha just said, some of the obstacles have nothing to do with getting paid. They may have to do with getting a ride or getting bus fare or getting, some taxi help or some way to get to the site frequently, or babysitter help, because if you go and the 3 kids are home and there's no one to take care of them, then you're not going to sign up for this trial. So reimbursement can be not just cash in your pocket, it may be to facilitate your getting to the trial site and making it possible to be a participant. And I would say, just as a rule, no, not so good at funding that kind of thing. There's not big pots of money out there that make that easy to do or doable. The overall funding structure, just so you know, is about 50-50 private industry versus the NIH. It used to be way more slanted toward NIH type studies, but private companies sponsor a great deal of research, and they have their own goals about who they want in their studies. For example, we've argued for many, many years that you need to get more children and pregnant women into studies. Many people look at that from the private sector and say, I don't know, that's a liability. I remember thalidomide long ago. I'm not sure we're going to take that risk, so we're just not going to do it. And it's very hard to twist an arm and say, no, you have to have women of childbearing age or women who you know to be pregnant and expose them to things that aren't yet FDA approved. So those can be significant hurdles. Sometimes companies will stop a trial because somebody else is ahead of them on the business side and people are left hanging. There's sort of compensation or the incentive to get knowledge out may end with different reasons. It happens in academia. People move, they get recruited away, they get in fights about where's the data going, where the subject's going. That's the real world of how that sometimes breaks out. So overall, funding streams tend to focus in three areas, and I'll end with this. One is who's talking the loudest to Congress about their problem. So it's always good to have a celebrity with a disease. People in Congress love that. And if you show up with George Clooney, you're going to get more money than if you show up with Art Caplan. So it's just the way they lobby and sort of how that goes. Second, the NIH funding, to some extent industry, really excited about mapping the genome, very excited about precision medicine. If I wanted to fund, why is it that we did so poorly in communicating information to the public in terms of marketing vaccines? I'm uphill. A lot more molecular biology, genetics than I'm going to see in the social science, much less on the prevention side of things. We have a very rescue-oriented industry and a rescue-oriented NIH for that matter. Then the last thing is we've seen a lot of emphasis because of this precision medicine in funding rare and ultra rare, like diseases that have 10 cases. So I was laughing when you were talking about recruitment. In some of the studies that we look at NYU, phase 1 is it because there's only 10 people. There's not going to be a phase 2 and there's never going to be a phase 3. So you have to start to rethink, how are we going to weigh evidence if we give 3 kids something and 2 kids get something a little bit more? Is how we're going to deal with that kind of evidence. So ultra rare is incentivized for funding within our system due to lobbying by different patient groups. And like it or not, that distorts the way traditionally evidence is accumulated because you're dealing with tiny subpopulations. And the problem grows the more we start to differentiate, say, cancer into genetic subtypes. You know, everybody could wind up as a rare disease down the road because the genetic variants of our disease, are whether it's depression or arthritis or whatever it is, you could see a lot of micro subdivision when people say, as NYU likes to say, but all academic centers do, we have personalized medicine. It's made for you. Well, that may be, but it means that it's hard to generalize from medicines that are made for a few people.
If I could add one thing to what was just said, we haven't really talked about human challenge trials as well, and I think we ought to identify them, and I'll leave that to the larger group. When it came to COVID-19, the government and Dr. Fauci and others decided there would be no human challenge trials, but England had them for the AstraZeneca vaccine.
And has them, still
has them, right. And it wasn't so much paying these people for the risk. It was these people were in quarantine for months and they basically had no income. And one of the problems that we face is that when you, especially in a stage 1 human challenge trial, you have to be very careful that the reward is not such that it will encourage people to go against what they may think are their best interests. In other words, that becomes a money-making proposition for them. So I would just throw that open to the group as well.
David, I just the communication person in me. So I don't know if everybody knows what human challenge trials actually are. Would you mind just giving
Sure,
like
Art,
that?
I'll leave that for Art.
Okay.
I mean, I know it well, but Art can crystallize it for us.
I mean, just very quickly, sometimes you have an agent and you want to see whether, say, a vaccine is going to work, but the disease you're trying to study is relatively rare. So if you tried to wait for natural infection to see whether the vaccine prevented it, you'd be waiting years because it's just a rare disease. The way to speed it up is to deliberately give the person the disease then see whether a vaccine that they've also gotten prevents it or not. So you're exposing people to potentially dangerous agents
Right.
with no intervention to save them. If we had that, we wouldn't be studying these things, we'd be
Right.
treating them. And it's quite controversial ethically. I came down on the side of doing them, but I certainly understand those who were hesitant. But that's what David's talking about, is
Yeah.
deliberately exposing someone to a disease in order to test more quickly a prevention or a cure.
Right. And the reason we turned it down here in the United States was that we had no remedy for the disease.
Although, again, by the
Right,
way, I'm
if the
sorry
person
to be
got it.
yammering here,
Yeah.
Kim, to shut me up, but the funny thing about the objection that you can't do something, give someone a disease when you have no remedy. If you had a remedy, you don't need to do a challenge study. The whole thing is completely circular. It's only for diseases where there are no remedies.
I agree with that. But the other thing I would say is that historically, human challenge trials have been used constantly.
Yeah.
Edward Jenner, who developed the first vaccine, the smallpox vaccine, used human challenge trials with children. And as most of you know, I'm sure that convicts played an enormously influential role in human challenge trials over the decades and over the centuries.
I think Pasteur too, right, with rabies?
Yes, absolutely. With anthrax, yeah.
Yeah. Okay. So, but maybe on the topic of this ethical challenge, I think we might make a move towards opening things up to a broader Q&A. So I'm going to direct one last question from me towards Julie Sowash. And then I think if the folks in the audience have questions, please type them in the chat, and Sharonik will offer them to the panel. So the last question from me is, what would be the statistical ideal of popular representation in clinical trials? What would produce the best research, and how far are we from it?
Well, I think, again, we kind of just start with that. We don't know how far we are from it because we don't collect the right data. And that's really the first step to addressing any issue is identifying what data are we going to collect. Ideally, the population in the study is going to be reflected of the population of the disease state. And so that we are able to ethically and efficiently test for a variety of different diversity types, ability types, et cetera. That's important. I think we all need to agree to that standard. And we also need to agree that we are not going to actively exclude people from underrepresented communities because of outdated, non-modernized stereotypes. And I'll stick with disability because that's my expertise, and over infantilization, over protectionism of a community who are able to consent and participate. And so ideally, we're going to have a population that looks like the disease state, that's going to be reflective of all of those different types of identities that are important and impactful to not just our physical health, but the socioeconomic determinants of health and the social determinants of health. And I think that's really where we need to start and just agree upon. And for us to really get to that place, we have to start with robust data collection that can be shared across studies and across therapeutic areas.
Hey, Kim, can I just jump in with one other comment? Because it's about, there's a fair amount of research that has been offshored, particularly to China. And so companies, but also universities, are studying various drugs, cancer most frequently, but some other things in China. And the question has arisen, one, can you generalize back to the US population, both in terms of genetics, environment, diet, economy, whatever, does it work, or is that just a cheaper, easier place to go? And then the other question that keeps coming up when you say, you know, I've talked to a number of friends of mine in China, and they say, well, are they doing the study in South China? Are they doing the study in Peking? Because we would never think of those populations as the same. You know, it's sort of like, here's data from China, but they would go into hilarity and say, oh, you mean one out of the 150 ethnic groups that we have? Which one did you use? And that's, as Julie was saying, that's not how it's reported. Nobody, it's just data from China, so to speak.
Mm-hmm. And just to build off those last two comments, I'm in various groups here at NYU, and we've talked about, okay, what's the right representation, right? That's like the $54,000 or whatever dollar question. And I'm always asking, is it based on the US Census? Is it based on New York City demographics? Is it based on the demographics of our health system and who we actually serve within our walls? It could be really challenging because there's so many ways that you could justify what the quote unquote right representation is, and it can really differ. If you're talking, say, about Black adult, well, are you talking about African Americans born and socialized in the US? Or are you talking about anyone who self-identifies as Black, which could include folks from, Africa and the Caribbean and all these different things? And I don't think we always have a great kind of answer of like, what is the right anchor of representation? What's the reference point? For that. It's a very interesting question. And there may be situations where you want to over represent or over recruit because historically those folks, are more affected. But in something like with disabilities, it's easier, I guess, technically to see like a physical, but there's so many hidden disabilities. And typically because people don't want to report that or it's not collected, for a variety of different reasons, there may be a situation where you could say, well, we need to over-recruit or have over-representation. And on one hand, I think that would be a good idea. And then you might have someone who's overprotective and say, oh gosh, we're being predatory. But I don't think it's being predatory because all types of people have health concerns and health issues. And when something is FDA approved, everyone in the population is going to get it, whether or not, most of the time, will have access to it, whether or not their patient population was represented in the trial. So the whole issue of what's enough representation and how you're deciding that, I think is a evergreen question that I haven't seen a good answer to.
And if I could just weigh in one more time, I think one good example we have seen recently of this is, I think it was 2022 earlier this year in April, CMS put out specific guidelines that a certain Alzheimer's drug could not have participants who had Down syndrome. And what we know is that 90% of the Down syndrome population is likely to acquire Alzheimer's over their lifetime. And for us to have a community that is specifically excluded when it is very reflective of the disease state is not okay. And fortunately, we had activists and advocates who were able to speak up and have that conversation, but we don't always have those voices. And so where we're seeing the data we can collect, we start there, but we can also look at the data about what are the non-scientific reasons that we're excluding people, and that's going to help us to get to a more reflective trial.
I think one of the things as well is that there seems to be a disconnect between the medical community to some degree and minority communities. You will have the medical community saying minorities don't want to take part in these trials. And you have the minority community saying we were never asked. And I was looking at some studies where they were talking about physicians and these trials. And a lot of white physicians said minorities like Blacks will not take their roles seriously. They don't follow instructions. They're more likely to drop out. And there's no evidence to support this. But it seems to me that what you have is this dichotomy where there are these stereotypes that sort of don't encourage minority participation.
I agree with that. I was going to say I think it's pretty well documented that although, medical mistrust and those things are real issues in racial and ethnic minorities and other groups, it's not always the predominant reason why people aren't participating. It's pretty clear that a lot of racial and ethnic minorities in particular, but you can extend that to older adults, 65 and older, are often not asked. They're never aware that these trials are even available. Now, I will say one, I think, step in the right direction, although it won't fix everything, is like places like NYU and other academic medical centers have moved to like an opt-out system where patients can be alerted of clinical trial opportunities directly through the patient portal and contact it directly via updates through the notice of privacy practices. And I think that's actually a really good thing. We had a lot of conversation at NYU, and we actually updated our policy in 2016, and some physicians, which I understand, were saying, oh my goodness, I don't want anyone contacting my patients. They're going to be confused. But on the other hand, when you rely on a gatekeeper system and only rely on physicians and nurses who are already busy, already overwhelmed, and when you're at an academic medical center that literally has hundreds of studies going on at any given time, it's really not feasible. And they don't have the time. So when you think about equity, that's one way to enhance equity, is that people at least have an equitable opportunity to know what their opportunities for clinical trials are, and then they can decide for themselves if they want to pursue it or get more information. But I think kind of removing that gatekeeper system is very helpful.
It's also possible that minority communities do not generally receive their medical care at major academic centers.
By the way, one other comment here in the four of us trying to pursue, well, what is the right ratio of whoever? The answer is there is no single answer, because different diseases and different disorders require different types of subject populations. I'm not going to study sickle cell in whites, and I'm probably not going to aggressively go after cystic fibrosis in Native Americans because we don't have much of it. But I may find that for arthritis studies, wear and tear arthritis, osteoarthritis as it's called, I could study it just in African Americans and generalize to everybody, because it's a wear and tear disease and it doesn't really matter so much who's the subjects. Whereas if I'm going to be looking at an area where there might be biological and genetic, ethnic differences to how people respond to a drug, or what the metabolism rate is, or what the comorbidities are because they're sicker in one group than another group, then it does matter where, what I'm trying to do. So I don't think there's an ideal. It isn't match the demography or sample the phone book, or, make sure everybody who lives in a town, some subsample is involved. Although since I'm from Framingham, where the heart study was from, that's exactly how that study was done. But, and I wanted to mention one other irony here, which is in some studies that I have been around over the years in transplant, when you take something like the one drop rule, it turns out that you're going to say certain organs that we get couldn't go necessarily to someone of a different race group. But it may also be the case that the immunology and blood types and biology of the one drop rule, it sort of undermine the fact that there are a lot of people who could take organs from white donors to quote unquote Black donors, or African American recipients. Similarly, with Native Americans, you go out and look at members of tribes and you see you want to recruit them to studies, but when you get them sort of biologically, genetically analyzed, they're more white. They're culturally Native American. They may even speak Native American languages, but that's not the history of what happened. Maybe if you're a quarter Oglala Sioux or Cherokee or something, then you can be the tribal leader, but it doesn't mean they're the best subject necessarily to represent quote unquote Native Americans. So it's thorny that way. It's hard.
If we go to the chat, I think there is
yeah. Sorry, David, to cut you off. Yes, I was just going to suggest that we go to the chat. We have one question. Most grants want pilot data to support, which can be frustrating to try and get pilot data in an area and make the pilot population diverse. Any recommendations on this from the panel?
I guess whoever is your funder for the pilot, I would ask them what they think diverse is. So like diverse is just a difference. And when I'm giving talks, I usually have this slide, we tend to default to diversity being race, ethnicity, and sometimes gender or sex, and sometimes age. But diversity could be different stages of a disease. It could be geography, it could be ability status, it could be SES, it could be a whole lot of things, literally tens of things. And so I think we need to be really careful of when we say we need diversity to say what you really want in that particular instance and why, because it may not make sense to say, okay, we need racial and ethnic diversity in this sense. Maybe the most important thing is a diversity of experiences or diversity of dyads or triads or whatever. So really, when we just use this as like a catch-all diversity, we're enhancing diversity. What does that mean? Just say what you actually are trying to do and why that is critical for that moment. But diversity is a lot of different things.
We do have a little bit of time left if anybody else in the panel wants to come at that question.
Well, what I see is the default for a lot of funders is what I'll call the census model. So are you representing your state, your nation, or the community? And if you're not doing that, then there must be something wrong. And I agree with Aisha, that's not necessarily at all the best way to start a pilot or anything else. And I do think you should ask, what sort of diversity are they thinking about? I mean, rarely does anybody say, well, are they left-handed or right-handed? Or what's their height? Could we study that? Or, how's the blood pressure variability in these subjects? Or what? I mean, there's a zillion ways to be diverse. We have our favorites, partly as David knows better than me due to history. But there are occasions when somebody wants to say, do you have implants? Are you someone who went through treatment as a child for something? And they're, so you've got to ask, because I guess I'd say this diversity, I'll be clever, is endless. I mean, you can go on and on forever with it.
Yeah. And I hate to call out my colleague, but hi, Doris. Doris Vieira is on the line. She's actually a medical librarian at NYU who is actually a co-I on one of my vaccine hesitancy related grants. And I will say, I think librarians, in addition to other health professionals, are really great resources because they do a lot of systematic reviews, do a lot of things related to different populations and who's been studied in the literature. And sometimes it's not helpful just to kind of continue doing studies on the same groups of populations that we've always done it on, working with someone like Doris or a medical librarian. You could say, okay, well, who's been historically left out and where are the gaps? Working with someone with real training in this area is a lot better than just doing a random Google search. So I will say, shout out to the librarians in the world and the biostatisticians, but I saw her online. It's nice to see you, Doris.
Amazing. I think the world definitely needs more librarians. So that's a really great note.
Absolutely. We'll be more literate and kinder and smarter.
Totally.
I think we also need...
I want to say, go ahead, Dave.
No, you go ahead.
We need more biostatisticians as well.
No.
No. Okay. All right. Well, at any rate, I wanted to thank everyone for attending tonight's webinar, and to say thank you to everyone who has attended our webinars over the past year. We are grateful to everyone, to our speakers and to our sponsors, without whom we could not have run this project. And I, in particular, am grateful to Shranik Bosu and Katie Grogan, who have been excellent colleagues and wonderful friends, who helped me transform a glimmer of an idea into a concrete reality that is here before us all this evening. So I'm really grateful. As we mentioned earlier this evening, this webinar is being recorded, and recording of this webinar and past webinars in our series will be available on the Gold Foundation website, which I will put the link for you guys in the chat. And we would really love to hear your thoughts on this webinar, so please don't forget to fill out our survey. Thank you so much.
Thank you.
Thanks, everybody.
Thanks, everyone.
Thanks.
Good night.
Thank you. Have a great night.